Good evening, everyone. Please confirm if I'm audible and the screen is visible to all of you. Yeah. So today we are back again with another round of question answer discussion of your CSIR NET December 2024. So I will be first discussing few questions both from your morning session and the afternoon session, both part B and part C. And later on, Tanushree ma'am will join to discuss other sets of questions. All right. So I hope you are all ready for this. Give me a thumbs up if you are all ready. So let's start the discussion. So first I'll start from the morning shift. Okay. The first question is the epigenetic regulator Demeter and Medi act synergistically to repress endosperm development in the absence of double fertilization during seed development in Arabidopsis. Which one of the following options is the correct enzymatic function of DME and MEA? Okay, so they have given the option DME is a DNA glycosylase, MAE is a DNA methyltransferase, DME is DNA methyltransferase, MEA is a histone methyltransferase, DME is a histone methyltransferase, MEA is DNA methyltransferase, and DME is a DNA glycosylase, and MEA is a histone methyltransferase. So for this, you need to understand what is this DME and MEA and how do they work. So basically, these two genes, they are, as it is already mentioned, that they are uh, having a role during double fertilization, right? So basically, this DME is functioning in the central cell, okay, and you have MEA, which is again going to be functional here in the central cell proliferation. All right. So together, this DME and MEA, they repress the endosperm development in the absence of double fertilization. So this DMEs, this is a this is a demethylation. Okay, demethylase. This is a DNA glycosylase. So this DME's demethylation, it likely activates very specific maternal alleles, and your MEA mediated histone methylation ensures the sustained repression of the paternal or other genes that would otherwise promote the endosperm proliferation. So both of these has a combined epigenetic regulation, and they ensure proper seed development. Okay, because they balance the activation and the repression mechanism. So that is why the correct answer is option 4. Now moving to the next question. Which one of the following is an example of character displacement? And the options they have given you is two sympatric lizards with similar morphologies consume the same insect. Well, this statement is showing you that they are consuming the same resource. Okay, so this is not character displacement. Two sympatric birds evolve distinct beak shape to consume seeds of different plants. So this is a case of your character displacement. Why? Because the character, that is the beak, okay, which was maybe same, Okay, now you have to understand that this beak size, this is designed as per the food they eat. Okay, so if you have two same size beak, that means they are consuming the same food, right? Now if there is if you're consuming two same food, that means what, there is competition. There will be a lot of this competition. To avoid that, what happens, one of the birds, they one of the particular species, they work on the minute differences that exist in their beak size, and maybe they extend their beak size or reduces the beak size. Okay, so what is happening, the character which is the beak size, it is getting displaced, okay, from the original size. It is getting modified, so that is why this is the correct answer. A predator evolves highly visual acuity to catch camouflage prey. This is adaptation. This is not character displacement. This is a case of your adaptation. Two plant species have similar flower shapes to attract the same pollinators. This is again, this can be an example of mimicry also. Okay, so this is the example of your character displacement. If you look at the picture here, the idea will be very clear. So you have two birds, okay, Geospiza fortis and Geospiza magnirostris, two species, okay. They both had large beak to feed on the large seas, okay. Now, what did Geospiza fortis do when there was drought? So obviously there was scarcity of food. So at that time, this particular species of bird, it worked on the beak size and it reduced the beak size so that now it can feed on small seeds. So automatically, what it has done is it has avoided, the competition, right, by reducing its beak size. This is an example of character displacement. Now, going to the next one. Tectonic uplift can alter the drainage patterns of rivers, thereby isolating populations of many species. Population isolation of this kind results from which one of the following? So here we can understand that there is a physical barrier, right? It has altered the drainage patterns of rivers. Okay, and because the drainage pattern, say suppose the river was flowing like this, okay, now because of the tectonic uplift, say the river has developed some distributaries, or maybe it is moving, it has changed its course in different ways, okay, because of which some different patches has been formed, so the population got isolated. So this is a case of your vicariance. Okay, so vicariance, uh, it refers to the splitting of the species by geographic range. Okay, it, it is involved in, as you can see, it is involved in the splitting of the species by geographical range. Okay, and results in some discontinuous population. And why does it happen? Because of some kind of a physical barrier. Okay, which can be a mountain, which can be a water body, in like in this case. All right, and because of this difference, what happens, the same species, when it has got divided into two different populations, it can evolve in its own way and become two distinct species. Now this is different from dispersal. Dispersal is like the habitat is not broken down, okay, the geographic space is not broken down, but rather the species from here chooses to migrate out, get and colonize some other space. Okay, and over here it evolves to form a different species. So this is a very distinct, there is a very distinct difference between vicariance and dispersal. Next, spindle assembly in animal cells requires nuclear envelope breakdown. Nuclear envelope breakdown is a multi-step process which begins when CDK1 cyclin B phosphorylates multiple components of the nuclear envelope. Given below are some components that are directly phosphorylated by CDK1 cyclin B. So here, this is a very straightforward question from your cell cycle regulation. So you just have to know what are the targets of CDK1 cyclin B. Now, as you know, CDK1 cyclin B, this is mainly is gets activated during the transition from G2 phase to M phase, right? Over here. So that means what? M phase is the place where the cells are going to divide, right? So therefore, what happens is during this G2 phase, the A type cyclins, which is mainly associated with the S phase, okay, this undergoes ubiquitin mediated degradation. And the B type cyclins, which are your also known as your M phase cyclins, they get actively synthesized. Okay, this will allow your CDK1 to bind to your two different cyclins, B1 and B2. All right, and this initiates mitosis. So therefore, this cyclin B CDK1 complex, it actually goes and phosphorylates almost 70 target proteins. All right. And because of that, it regulates many key mitotic events also, like centrosome separation, chromosome condensation, nuclear lamin breakdown, Golgi disassembly. All right. So therefore, over here, the correct answer is option 3. Nucleopore complex is one of the target components. Another one is a nuclear lamina. Okay. Now, moving to the next question. So this is a question from your bacterial growth curve. So there can be different kinds of growth curves seen. Okay. So it can be a continuous, it can be asynchronous, synchronous, diopsic. So let us understand what these curves they mean. Okay, the correct answer here is obviously option 4. The first one is synchronous. The second one is asynchronous. C is continuous, and D is your dioxic. But what is the meaning of all these? So basically, your synchronous culture. This is this refers to the growth process where the individual cell shows synchrony with the other cells. That means they grow at this, they have the same growth phase. So one cell is growing, okay, then there is a station, there is a stationary part. Again, the cell grows and it's the same amount of growth. Okay, same amount of growth. Again, there is a stationary part. Again, same amount of growth. Again, a stationary part. Okay, so that follows a synchronous way of growth. Okay, so same growth phase for a given generation time. That is synchronous. So, okay, now what is your asynchronous? Asynchronous is the most commonest type of bacterial growth that we get to see. Because any bacterial growth has a lag phase, then a log phase, then an exponential phase, and then a stationary phase. So here, as we can see, the phases, they are not same, right? The growth rate of in these phases, they are not same. So therefore, this is your asynchronous. All right, asynchronous growth. In the case of your continuous growth, this is seen in the case of continuous cultures. Okay, like in the case of your chemostats, where you are maintaining the bacteria for in a log phase by constantly replacing the media. So obviously over there you will see a very constant growth pattern. Okay, so this is continuous. And then you have the dioxic growth, which is also known as your diphasic bacterial growth pattern. And this you will get to see when you expose the bacteria to two different carbon sources. Like you have read in the case of your lactoperon, right? When glucose is present, lactoperon doesn't get switched on. But when glucose depletes, then the lactoperon gets active, okay, and it starts utilizing lactose. So in this case, we will get to see a dioxic curve. a curve where you will see that yes, the glucose utilization was happening, and then there is a stationary phase where glucose has depleted and lactose needs to be utilized. So you need some time here, again some lag phase is required, and then again you will see the growth is coming. Okay, so this growth is for the breakdown of lactose, this growth is for the breakdown of glucose. That is your diopsic curve. Next question. So then we have is saying the helicase activity of E. coli DNA B was investigated using the following two substrate 1 and 2. Okay, under various conditions. So these are the conditions, followed by gel electrophoresis and autoradiography. The results of these experiments are depicted below. So they have, so the results are given here, and what kind of substrate you have used is given here as well. Okay. Now, what is the basic difference between the two substrate? In this case, you see that the replication has not started yet. Okay. But in this case, these forks clearly demonstrate that the hydrogen bonds over here is broken, and a replication fork has already been constructed. Okay, that means helicase is there. So the hydrogen bonds have been broken. There is a free end, free 3' end, but over here it has not started yet. Okay, this is a basic observation that you can make. Now you have to analyze these bands. Okay, what they are representing, and each band corresponds to each of these combinations, the conditions which are provided here. Okay, so now you have to analyze these conditions and you have to analyze the bands. So here you can see, like, when you are comparing, like when you compare this and this, see here, these two columns, if you see, you have SSB in both the columns, DNAB in both the columns, ATP in both the columns. Okay, now what is this DNAB? It is your helicase, right? It is already mentioned, see? And even if it is not mentioned, you should be doing that DNAB is your helicase. You also have ATP, and they have given the boiling, which is negative for both. Now, since all the conditions are similar, you should have got same band pattern, but what you have got is different band patterns, right? So over here, this band is telling you that there are some fragments, smaller fragments, which has migrated in that given time. The fragment has migrated. That means if it has migrated below to this part, okay, to the down part of this electrophoretic gel, that means these are smaller fragments, okay, as compared to this one, because obviously the smaller fragments, they can migrate faster than the larger ones. So you can say that these are the Okazaki fragments, right? We can assume it. So when will this Okazaki fragments begin or be produced? When there is replication, and particularly the lagging strand. Okay, so here you will get Okazaki, from here you will get Okazaki, right? Next, why you are getting it? Because here obviously the DNA is unwound. Okay, so from here we can say that statement 1 is correct. Okay, DNA we can unwind only a partially unwound DNA. So here the DNA is not unwounded. Okay, but here it is. So your helicase will further unwind the DNA. Next, if you compare this column, this column, and this column, now what is missing in this column is ATP, right? And because of that, we see that the fragments has not migrated at all. It is there where it was. Okay. So from here we can conclude that ATP is required for DNA unwinding, because otherwise, see, both the columns are same, right? Except for the fact there is ATP missing. So ATP is definitely required for unwinding. All right. So that is why option 4 is correct. Next, lethally irradiated mice were split into 4 groups, and experiments were conducted as given below. Group 1 was not given any cells. 2 was given thymus derived cells from syngenic donor, and 2 months later immunized with polysaccharide antigen. Group 3 was given bone marrow cells from a syngenic donor, and 2 months later immunized with a polysaccharide antigen. Group 4 was given bone marrow cells from a synergic donor, and 2 months later immunized with a T dependent antigen. Okay, so there are 4 possible outcomes predicted. You have to find out which is the correct option, right? So let's understand this. In the first group, you have this lethally irradiated mice. Okay, now this mice will not be able to survive. Okay, they will not be able to survive long term without a bone marrow transplant. Okay, so without this treatment, they will die within 2 years from this because of hematopoietic collapse. So immunization is irrelevant, and obviously no immune cells exist to mount a response in this case. All right. So that is why this, this mainly tells you that it is necessary of bone marrow. I mean, bone marrow is necessary for sustaining life and immunity after lethal radiation. Fine. Now moving to group 2. In group 2, mice, it will fail to mount an antibody response to the polysaccharide antigen. Why? Because there is absence of B cells. Okay, since there is no uh, bone marrow reconstitution, so there is absence of B cells, and that renders the immune system incapable of producing the antibodies. Okay, despite uh, the fact that there is presence of your synergic T cells, right? So from here we understand that this B cells, it plays a very critical role in antibody mediated immunity, and even for your T independent antigens. Fine. Next, group 3. What will happen to group 3? Here the mice will mount a T independent antibody response, which is mainly dominated by your IgM. Okay. So it will give some transient protection, but obviously it will not be a very lasting memory. In the case of the fourth group, okay, in the fourth group, here the mice will generate a very strong antibiotic response. Okay, there will be a class switch antibody response. Here we will see that it is mainly IgG which dominates. Okay, so first you get IgM and then it shifts to IgG. Okay, which has a long-term memory, and therefore it tells you that this is a very successful immune reconstruction constitution, and you get a very functional T cell here, and that helps to uh, helps the mice to survive. So therefore, the correct combination here will be statement A, group 1 mice are unlikely to survive. Statement C, C are likely to produce antibodies in response to polysaccharide. D also, they are likely to produce antibodies, okay, in response to T cell T dependent antigens. So option 4 is correct. Moving to the next one. Here they are saying that pollen tube in growth in transmission tract of style and its attraction to the embryo sac are directed by chemical cues in plants as discovered in lily and torrenia. Which one of the following statements about the proteins and or peptides secreted in the process is correct? Okay. So basically, when pollen tube grows, as you can see, the pollen it falls on the stigma of the gynecium, and then from the the pollen tube grows. Okay, and carrying with it the two sperms, and it goes and fuses with the ovule in the ovary. So here, the synergids which are present in the ovule, okay, they secrete a cysteine-rich protein like your LURE protein. Okay, and this attracts the pollen tube towards the ovule. Next, you have the epidermis. It secretes the cysteine-rich adhesins. Okay, like your SCA, which is a six stigma style cysteine-rich adhesins, and also a chemocyanin. Okay, particularly in lily, you will get to see that there is a chemocyanin which is released. So this create a very supportive microenvironment, and it also helps and it also guides the pollen tube growth. Okay, so these proteins or peptides, they are uh, like conserved, yet their very species specific molecular mechanisms exist to guide the pollens, but these are very common proteins which are used. Okay, so therefore the correct answer here is statement 2. Now, moving to the next question. So you have RuBisCO. Okay, RuBisCO enzyme catalyzes carboxylation or oxygenation of RuBP in 5 steps. Following a certain statements regarding the catalysis carried out by RuBisCO. So statement A says the first step of catalysis is enolization of RuBP. Carbon-carbon bond between C3-C4 of RuBP is cleaved. Carboxylase activity produces only one molecule of 3-phosphoglycerate. Oxygenase activity produces one molecule of 3-phosphoglycerate and one molecule of 2-phosphoglycerate. Which of the following options is correct? Okay. So mainly, if you see, this is a summarized picture of C3 plants, SCAMP and C4. So we will mainly focus on C3 and C4. Mainly, there are 5 steps. Okay, the first step is activation of RuBisCO. So what happens in this when the RuBisCO gets active? A CO2 molecule will bind to the lysine residue in the enzyme. Okay, fine. This reaction obviously requires a magnesium cofactor, and that results in the enzyme becoming catalytically active. In this case also the same thing happens, even though the RuBisCO is placed in the bundle sheet and not in the mesophyll cell, but still you have the RuBisCO has to fix the CO2. Okay, so same process happens here as well. The next step, so this is step number 1. Okay, activation. It is activation. Step number 2 is binding of your RuBP to the active site RuBP. Okay, so this binds to the active site of your RuBisCO. Binding happens. Next, number 3 is enolization. Okay, enolization of your RuBP happens to form an enodiolate intermediate. Okay, so this RuBP adopts an enodiolate intermediate structure, and that step is your enolization. Number 4 step is carboxylation. This is the number 4 step. Okay, which is in very simple terms is addition of CO2. So that leads to the formation of an unstable 6C intermediate, okay, or oxygenation, leading to different intermediate of CO2. Okay, now this 6C intermediate, it splits into two molecules of 3 PGA, which is a 3C carbon. All right. Now that is the fourth, and the last step is a cleavage of the intermediate. All right. So this cleavage of the intermediate into the respective products happen. So in carboxylation, 2,3-PGA molecules are formed, which is used in Kelvin cycle. Because of oxygenation of your RuBisCO, 3-PGA and 2-phosphoglycolate is formed, which enters your photorespiration. Okay. So therefore, the correct answer here is option 4. Moving to your next question. A researcher simultaneously inhibited the activity of triose phosphate translocator and xylulose 5-phosphate translocator in a plant and made the following assumption. So what are these translocators? So here that it is present. Okay, this is your this is the cell, this is cytoplasm, and here you have the chloroplast. In the chloroplast, you have this triose phosphate translocator present. Okay, so mainly this triose phosphate translocator, which is mainly embedded in the inner envelope membrane of the chloroplast. Over here, there will be no sucrose synthesis if this particular inhibitor is, if this translocator is inhibited. Okay, and you have another translocator, which is your xylulose 5-phosphate translocator, which is also located in the inner chloroplast envelope. That basically links the cytosolic oxidative pentose phosphate pathway to the chloroplast Kelvin cycle. Okay, Kelvin cycle, and that helps in the regeneration of your RuBP. So if you inhibit the XPT, okay, it will mainly cause the Kelvin cycles to stall because of your RuBP shortage. RuBP will not get generated. Okay? So therefore, over here the correct option was option 2. Statement A is correct. Triose phosphate will accumulate more in chloroplast, because as you can see here, it is not going to get transported out to the cytosol. And D, xylulose 5-phosphate will accumulate more in cytosol, because obviously it will not be able to pass it to your this oxidative pentose phosphate pathway. Okay, so 2 is correct option. Okay, so far, do you have any doubt? No doubt, right? Okay, fine. So let's continue. Now coming to the next one. A parasitoid infects a host to complete its life cycle. Which of the following life history traits typically characterizes this parasitoid, assuming that only one parasitoid infects one host? Very interesting question. So let's first understand few things before we get into the statements. There's something known as your intrinsic growth rate. Okay, see, they have used this word intrinsic. So this intrinsic rate of increase, it mainly means the maximum potential growth rate of a population under any ideal conditions. Okay, so for parasitoids, which are like insects whose larvae develop or the host or maybe inside the host, and they kill the host. Okay, so their growth rate must be obvious, must be closely tied to the host population dynamics because they are so much dependent on the host. So if the parasitoid's intrinsic growth rate, which is denoted as R, okay, small r, it is similar. It suggests that they can keep up with the host population growth, which is very important uh, for biological control. Now, if your parasitoids R is low, okay, they might not control the host effectively. On the other hand, if it is high, they would overexploit and crash the host population, leading to instability. Now, as per your Lotka-Volterra model, we have studied that your parasitoid success, it depends on the ability to find the parasite's host, right? Host parasite interaction. So the factors like fecundity, development time, survival rates, they are all key. Okay. So host might have higher intrinsic rate because they can reproduce more quickly, but parasitoids often have strategies to match to match. Okay, like shorter generation time or higher reproductive rates. So therefore, if you now read the statements, you will see that option 3 is correct. So intrinsic rate of population growth comparable to host, eventually fatal for the host, because why? The parasitoid is capable of maintaining its growth rate with the host, and that is how its life cycle is also designed. Okay, now let's come over to a question from ecology, community ecology. So they have given you three communities. Okay, one is telling you that it is present, that particular species is present. Zero is telling you that it is not present. Okay, and they are asking you to find the Sorensen's coefficient of similarity. So basically, this is exactly not from your ecology portion, but rather this is from your unit 9. Okay, where you study the Jacquard's coefficient, and there are other coefficients also. So Sorensen coefficient falls under unit 9, part A. However, let's understand what it is saying. So how do we calculate? First, we need to understand how do we calculate. Okay, the formula is Sorensen's coefficient of similarity is equals to calculated as 2a by 2a plus b plus c. Okay. Now what is this 2a? This into 100. Okay, what is this A? This is actually the number of species which is common to both. Okay, so the two species that you are, the two communities that you are comparing, you have to see which all are common. So you have to calculate the number of commons common to both. Okay, B is a number of species in the first sample. So in the first sample, the number of species which are there. Okay, and C is the number of species in the second sample. Now, what do you mean by second sample and first sample? It is the first community and the second community. Okay, that way also you can write. So now let us compare. Okay, if I take xy, what am I getting common to both? Will be 1, 2, right? Only, so that is my A is 2. Next, if I take B, how many present? 1, 2, 3 is present. So my B is 3. In the case of Y, how many present? 1, 2, 3, and 4. So C is my 4. Okay. So I will just put this information in the formula and I will calculate the Sorensen coefficient for X and Y. Similarly, in this same way, I will calculate for your XY and you will see it is coming to around 22%. XZ, which is coming to around 20%, and YZ, which is coming around 18%. Okay? So from here, you can say that the second option is correct. Okay, XY greater than XZ greater than YZ. All right? Okay. Quickly moving to the next one. Which statement about studies on succession is incorrect? The correct answer is 4. Succession studies show that co-evolved native species always outcompete invasive species. Okay. Next, which one of the following statement is not part of the neutral paradigm? Now, what is this neutral paradigm? Neutral paradigm, it is actually talking about the neutral theory of molecular evolution. Okay, this mainly suggests that most evolutionary changes at the molecular level, they are all due to your random genetic drift of the neutral mutation rather than natural selection. Okay, that is the that is a principle of a neutral paradigm. So according to that, statement 4 is correct. Ecological drift results in stable coexistence of a given set of species. Fine. So that goes with your morning shift. Now let's move into the afternoon shift. So the first question here that I have from the afternoon shift. All right, before I get into the afternoon shift, I hope this part is clear to all of you. Don't have any questions. Right? Okay then. Then let's continue with the afternoon shift. So what is the correct order of enzyme actions during long patch base excision repair in humans? So for long patch base excision repair, you have to understand the basics of your base excision repair and then understand what is a long patch. So this is your summary of the base excision repair, short patch and long patch. Okay. So what happens here is mainly this long patch base excision repair. This addresses the damaged bases by excising a gap of around 2 to 10 nucleotides, okay, which is then obviously and filled and ligated, okay. Now this involves the excision of a gap of 2 to 10 nucleotides. So primarily it occurs in the proliferating cells and it utilizes the replication proteins for your DNA synthesis and gap filling. Okay, so that will include your DNA polymerase delta and epsilon. It will also include your pCNA. Okay, so what happens is the damaged base is first recognized. Okay, the damaged base is first recognized, and it is removed by your DNA glycosylase. All right, this creates the apurinic site. Now, an AP endonuclease cleaves the DNA backbone at this apurinic site or abasic site. Okay, so a gap of 2 to 10 nucleotide is created here, basically. Now, this gap is filled in by polymerase delta epsilon, okay, and the DNA strand is then ligated. All right. So, therefore, the correct answer here is statement 2. Glycosylase, epiendonuclease, DNA poly delta alpha, flap endonuclease 1, and then you have DNA ligase 1. Moving over to the next question. Which one of the following statement is incorrect regarding pattern formation during embryogenesis in Arabidopsis? Okay, so first let us look into the pattern formation that we have. So here the zygote, it mainly divides asymmetrically, as you can see, to give a smaller apical cell, and which is mainly the embryo proper, okay, and the larger basal cell, which is the suspensor. Okay, so this division, it establishes the apical basal axis of the embryo. Now, the octant stage proembryo consists of 2 tires, okay, each of 4 cells. Now, the upper tire gives rise to the apical region of the embryo, okay, from which your shoot meristem is going to be formed. All right, shoot meristem and the cotyledons also, they originate. Whereas the lower tires, okay, the lower tires, it produces a central region of the embryo which generates the remainder of the cotyledons and the hypocotyle. Okay, this part. And also it gives rise to the hypocotyle, and then it gives rise to the root meristem. Okay, so this is the developmental phase. So therefore the correct answer here is option 1. The zygote is unpolarized with respect to its intracellular composition. Now see, they have asked for incorrect statement. Okay, so obviously it is polarized. We have clearly seen that there is a apical basal axis which is formed. Okay, now moving to the next question. So they are saying that which one of the following statements best illustrates a dominant negative mutation? Okay, so what is this dominant negative? First, let us understand this. So dominant negative is a term which is actually used to describe a type of genetic mutation. Now this genetic mutation, this results in a protein which interferes with the normal function of the wild type protein. All right. So when will this occur? when you will have the mutant protein product, which can be non-functional, when this product competes with the standard protein product for binding to other cellular components. All right. So therefore, option 4 is correct. The mutation in a structural protein genes produces an altered protein that can interact with its wild type counterpart and disrupt its function. Very simple. If you just know what is the definition of dominant negative, you will be able to answer this type of 4 marks question. Next, this is a 2 marks question. Again, a very straightforward question from your embryocyte development. So, which one of the following plants has a bisporic type of embryocyte development? The answer is Arium sativa. Okay, so these are the different types of embryonic development. You have monosporic, uh, bimitotic, okay, you have monosporic trimitotic, you have bisporic bimitotic, tetrasporic bimitotic, and tetrasporic bimitotic. Okay, so that results in the different kinds of your embryosac development. And as you can see here, the main problem arises is in the division. Can you see the division part, right? Next, the presence and abundance of a species in local community is dependent on multiple processes. Which one of these processes is unlikely to depend on intra or interspecific interaction? All right. So dispersal is definitely based on intraspecific and interspecific interaction. Okay, there are different kinds of dispersal. It can be uniform, random, okay, or it can be clustered, and it is all very inter or intraspecific interaction. You will get to see. Niche differentiation is definitely interspecific interaction happens, because of which the niche gets differentiated. Okay. Now, what is this demographic stochasticity? This actually refers to random variation in population dynamics, which arises due to any chance event in individual birth and death rates, and because of that, it leads to a very fluctuation in the population size, which is very prominent in the case of your small populations. So therefore, this is not at all dependent on interspecific or intraspecific interaction. It is all dependent upon some chance event. Okay. So the correct statement is option 3. Next, a desert animal plant with long duration seed dormancy germinates only after heavy rainfall. What life history trait does this illustrate? Well, this is definitely not talking about case strategy. This is definitely not talking about frequency dependent reproduction, because there is no question of frequency dependence. This is also not talking about density dependent reproduction, because this is also there is no information on density, right? It is only saying that the seed dormancy is there and it germinates after heavy rainfall. So this is a case of your bet-hedging strategy. So this bet-hedging, this is actually a risk spreading strategy, which is used by organisms in a very variable environment. Okay, so in this case, in this uh, case, some long-term fitness across the generations is maximized at the expense of your reduced fitness within the generation. So here you are not producing the seeds immediately. Okay, you are waiting for a heavy rainfall to produce the seeds, to germinate the seeds, not produce, sorry, germinate the seeds. So that means what? You are ensuring a long term sustenance, right? And not an immediate result. So this involves producing offsprings with different characteristics to increase the chances of survival in different environmental conditions. Okay, moving to the next question. For expression of gene of interest and a green fluorescent protein in mammalian cells, gene of interest and GFP must be expressed in a single mRNA but translated independently. Which one of the following would be the structure of the expression construct? Okay, so here you are transcribing it together, but translating it separately, right? So that means if this is your gene of interest and this is your GFP, that means here you should have this IRES, because otherwise the ribosome will not be able to bind separately to translate this GFP. Okay, and in the end you should have the PA, right, to ensure that the entire translation is over. I mean, sorry, the transcription is over. Okay, so that the mRNA is actually stable, you keep the poly A tail at the end of the whole transcript. And here you should have the promoter. The promoter should be controlled by the enhancer where the transcription factors will bind. So this is actually the transcript that you should have, okay, the construct. So therefore, this is the correct answer. Okay, moving to the next one. The region of a mouse chromosome was subjected to micrococcal nucleus hypersensitivity analysis over stages of development. In early stages, the region had irregularly spaced nucleosome. In later stages, the nucleosome were irregularly spaced with several nucleosome-free regions detected. Based on the above observation, which one of the following is the best possible inference? Well, you know, the regions of your chromatin, they are conditionally silenced, like how you will get to see in your X chromosome in females. Okay, only one X is active. But unlike your constitutive heterochromatin, facultative regions, the facultative heterochromatin regions, they retain the plasticity and they can switch between active and repressed stages. Okay, so facultative heterochromatin, most of the time you will get to see very tightly packed nucleosomes. Okay, and when that is the situation, it obviously reduces the DNA accessibility. But then again, the nucleosomes may adopt irregular spacing over the silenced genes, okay, blocking the transcription factor binding to these genes. So that is how the facultative regions are formed. Okay, so therefore, based on this observation, you can conclude that the chromatin is a facultative heterochromatin. Okay, so these are my sets of questions that I had. I hope you have understood these questions, right? So that's it from my end. Thank you very much. I will pass the controls over to Tanushree. Thank you.
So thank you, Somrhita ma'am, for such a wonderful explanation of the CSIR NET December 2024 questions. So I hope everyone is paying attention to the explanation part. So now it's time to discuss the questions based on unit 7, 9 and 13. So hello everyone, I'm Dr. Tanushree, and already in part one, part one of CSIR NET December 2024 question paper discussion, a lot of questions have already been discussed. And today also in part two, some of the questions have already been discussed by Somrhita ma'am. And now I will be discussing few questions from Unit 7, especially Part B questions I will be discussing. And both Part B and Part C questions from Unit 9 and 13 I will be discussing. So I hope you all are excited to see the answers and explanation and to check whether the answer you have marked in the exam is correct or not. Yes? Please type in chat. So we will begin with Unit 7, Part B Unit 7 questions. So here you can see this question, if you go through the question paper, the Part B questions, Unit 7 Part B questions, they were very direct. They were all memory based, right? It's just that they requires conceptual clarity. So if you are already thorough with the concept and you have properly revised the concepts, I'm sure you would have answered it correctly, right? So the question is, which one of the following is generated in skeletal muscle cells by single quanta of acetylcholine released from motor nerve terminals? See, such a very direct question. We all know that when a single quanta of acetylcholine is released from motor nerve terminal in skeletal muscle, it causes generation of miniature and plate potential. In short, we call it as MEPP. So MEPP refers to quantal and random spontaneous potentials recorded in a muscle cell. And hence answer is option 4 here. So let me know in the chat how many of you have answered it correctly. Then moving over to another question. Which one of the following statements about HCG, that is human chorionic gonadotropin, is incorrect? Now, we all know that CSIR, they specially use these terms to confuse you. And if you don't focus on these terms, okay, these terms are very important: correct, incorrect, true, not true. Okay, which of the statement is not correct or correct. So if you miss these terms, then definitely you will, even knowing the answer, you will mark it incorrectly. So I hope you all have focused on these terms, because during the tips and tricks session also, we always say that you have to focus on these terms, right? So HCG, the question is about HCG. So yes, we know it contains galactose and hexosamine, and its alpha subunit is identical in TSH. Beta subunit is smaller. No, this is incorrect, because we know the beta subunit is not smaller, it is larger. Okay. If you see the amino acid composition, then like beta subunit consists of 145 amino acid in comparison to alpha subunit, which contains only 92 amino acid. So definitely B subunit is incorrect, and hence it is a answer. Then coming to another question. Which one of the following is not a vitamin K dependent blood clotting factor? Another very direct question, right? So we all know factor 2, 7, right, 9 and 10. So these are all basically the vitamin K dependent blood clotting factor. The one which is not is factor 5, and hence it is the answer. Then which one of the following is not called secondary bile acid? Again, this is a very direct question. Bile acid, we have primary and secondary. So when you talk about primary bile acids, then it includes the cholic and deoxycholic acid. And secondary bile acids include deoxycholic and lithocholic acid, right? So basically, based on the given options, we can clearly say that, okay, this is primary, and even like also deoxycholic acid is also it is the secondary. Okay, this is secondary. And even we have deoxycholic acid and lithocholic acid as secondary. So since the question is which of the following is not the secondary bile acid, it is option 3, and hence it is the answer. Then which one of the following does not increase airways resistance in lungs? Again, another very direct question, memory based question. So if you have studied about all 4, So we all know thromboxane, histamine, leukotriene, these cause bronchoconstriction. They increase airways resistance. But the question is, which of the following does not increase airways resistance? So it is norepinephrine. So here answer is option 1. So see, these are very easy questions, right? They are not taking too much time, right? Then the order, now this question is from unit 9. So now coming to unit 9, part B questions. So the order Silotales can be identified by which one of the following characteristics? Now, you have to just recall here the characteristics of Silotales. And if you have, so especially those which are having a botany background, I'm sure you all have answered this question correctly, right? So if we read the characteristics of Silutaceae, we know that they are distinct in being rootless and they have dichotomously branched stems, which is given in option 1. And even some more characteristics are there, which I have mentioned on the screen. You can go through it. So this is a leafless and rootless body with a dichotomously branching stem is the characteristic feature which can help identifying this order. And hence answer is option 1. Then Sarah hardwicki, a spiny-tailed lizard, is a diurnal ground-dwelling species currently known from the Indian subcontinent. It is endemic to which one of the following habitats? Every year there is a question either on endemic areas from Unit 9 or migratory bird species. So this year also there was a question on migratory bird species, but even like they ask you about endemic areas, or as mentioned over here, they ask about this particular species, like it is endemic to which one of the following habitats. So Sarah hardwicki, basically, I'm sure some of you might have already know about it. It is known as Indian spiny-tailed lizard, and it is found in patches across Thar Desert, Kutch, and even surrounding arid zones of India and Pakistan. So this is a very direct question, and the answer here is option 4. Then another question from Unit 9. Which one of the following morphological characters can help you differentiate leptosporangiate ferns from eusporangiate ferns? So again, I expect the botany background students would have answered it correctly, especially. And even like for even from other background like biotech or microbiology or biochemistry, if anybody, if you have studied little bit of Unit 9 and about plant classification, and if you have focus on these terms, this question is very easy and direct. Like you don't have to dive in details of these ferns. Okay, like if you have studied about ferns, so leptosporangiate is one of the 4 major groups of ferns. Okay, and this name, the ferns leptosporangiate itself, like it is named because this sporangia arises from single epidermal cells. So every time, whenever, especially you are studying some terms, so just understand why so and so is called with so and so name. Right? So here the leptosporangiate, we call it as because sporangia arise from single epidermal cell and not from a group of cells as in eusporangiate. So that means the morphological character which is used to differentiate between these two is the number of cells from which sporangium is formed. So in leptosporangiate, it is from single epidermal cell, and in case of eusporangiate, it is from group of cells. So hence answer is option 3. Then which one of the following features now distinguishes echinoderms and cnidarians? So again, here you don't need too much detailed information. Okay, like you don't have to learn what all organisms belongs to so and so group. if you have just learned the or have gone through the basic characteristics, I'm sure you would have answered it correctly. So here in this case, we all know there is, like, if you talk about the classification, animal classification, one of the criteria for classifying is the germ layers, uh, the germ layers, right? So if you talk about echinoderms, they are like if you talk about echinoderms, they are triploblastic. That is, they have 3 germs layer. So based on germ layer, we can group the organisms, right? In animal classification, we use this one of the criteria. And in case of cnidarians, they are diploblastic. So triploblastic means that 3 germ layers are there. Ecto, endo. So ecto is there. Just a minute. So ecto is there, and meso is there, and endo is there. But in case of cnidarians, it is only 2. That is ecto and endo. So here answer is option 3. The total number of germ layers present, on that basis, we can distinguish echinoderms from cnidarians. Moving over to another question. So this time the question was on lectotype designation. So in unit 9, you study about different type, species type, right? Type species, holotype, lectotype, syntype, isotype, paratype, right? So if you are familiar with the term lectotype and you know what all are the rules which are being used for designating a lectotype, you would have easily answered this question. So if you are not familiar with this concept, I have included some of the rules which are being needed for lectotype designation. So always remember, isotype should be chosen for lectotype designation, and isotype must be chosen, or otherwise a syntype. So that means isotype is given a preference over syntype. And if we look at the given option, it is mentioned in option 1, a syntype is preferred over an isotype. And in option 2, it is an isotype must be chosen over a syntype. So these are contradictory. And based on this rule, which I have told you just now, obviously option cannot be 1, right? An isotype must be chosen. Now, if we look at the question is here we have to choose incorrect statement, not the correct one. So definitely 1 is incorrect and it is the answer, right? And further, if you want to confirm 3 and 4, so for your information, you can note down that yes, option 3, it states if syntype or isosyntype and isotypes are not available, paratype can be chosen. So this is correct. So lectotype must be chosen from among the paratypes if such exist. Like if no isotype, syntype or isosyntype is extant, then in that case, you can choose from the paratype. And if no cited specimens exist, then lectotype can be chosen from uncited specimen or of any original material as mentioned in option 4. So that means 2, 3 and 4 are correct. Incorrect statement is option 1 and hence it will be the answer. So you have to be very much particular about what is the, like what is being asked in the question. Is it a correct statement asked or incorrect statement asked? Then another question is, which one of the following is true for a monocot root? Again, not like you just have to in plant classification, when you study about the angiosperms, like when you talk about like if you have studied what is the basis of classification, okay, when you talk about thallophytes, bryophytes, pteridophytes, gymnosperms, angiosperms, and particularly when you study about monocot root, dicot root, so this particular diagram you just have to recall. So we very well know that monocot root is characterized by presence of epidermis, endodermis, pericycle is there, pith is there, which is large over here and well developed, and vascular bundles are there. And vascular bundles are often polyarch. Polyarch means when there are more than 4 vascular bundles. So usually this term is used when there is more than 4 vascular bundles. So yes, These are the characteristics, these are true for a monocot root. So pith is large and well developed, which is given in option 1 and 2, because in option 3 it is given pith is small or absent, and in option 4 it is given pith is absent, so obviously not correct. And basically vascular bundle is polyarch and not hexarch. So therefore answer is option 1 here. So these are all memory based questions. Now coming to part B questions of unit 13. Which of the following molecular marker techniques uses a single primer in PCR reaction for identifying polymorphisms between genotypes? Now, kindly note this question, you can keep it in unit 12 also, because in molecular markers you study in unit 12 also. Even in unit 8 you study about molecular markers, right? And even in unit 13 also you study about molecular markers like RFLP, AFLP, RAPD, right? Now, definitely to answer this question, you should be familiar with the working principle of these markers, molecular markers, right? So remember SSR, that is simple sequence repeat. So here we need a primer which is flanking the satellite region, microsatellite region. And AFLP, yes, it involves restriction digestion followed by PCR amplification, but multiple primers we use in AFLP. Okay, so I'm not going in detail of these molecular marker techniques. Okay, in class definitely we cover all these molecular markers in detail. So if you have studied about it, you know that in RAPD definitely we use short primers which are around length of 8 to 10, and RAPD, it is the PCR based marker where random primers are used, right? Not sequence specific primers are used, and yes, only single primer is needed. Although combinations like we use more like single primers are used, but like we use different sets of primers for every reaction. And SCAR is where which is derived from RAPD, and but here also two primers are needed. So because in the question only it is asked like which of the following technique only single primer is used, so it is RAPD, and hence it is the answer. Then coming to hormone pregnancy tests, another question based on immunoassay, or you can keep it in unit 7 also. So hormone pregnancy test works by detecting the presence of hormone, human chorionic gonadotropin, using immunoassay. Which type of cells will develop to produce HCG in pregnant females? Now if you have studied about this hormone, so basically it's a chemical which is created by trophoblast cells. Okay, trophoblast tissue, which is typically found in early embryos, and which will eventually be a part of placenta. So measuring HCG levels is very much helpful in identifying normal pregnancy, even in identifying pathologic pregnancy, and also can be useful following aborted pregnancy. Okay, so again, this question is very direct. So answer here is option 2. So the type of cells which develop to produce HCG in pregnant female is trophoblast cells. Then, which one of the following techniques can identify acetylation on lysine residue of a protein? So, see, every time, always I mention that when you're preparing for unit 13, you have to, like, if you're studying spectroscopy, you have to study all the spectroscopy. If you are studying microscopy, you have to study all the microscopy. And like in the exam, you get combination type questions where they give you an application based question, and in the options, they give you a combination of different techniques, right? And this is one such question. So question is very direct, that which of the following technique can be used to identify acetylation on lysine residue? Now we very well know that it's the mass spectrometry technique which is used to study the post-translational modifications, and especially when there is an acetylation happening, there is an increase in mass of 42 Dalton. So not acetylation, you can study phosphorylation also. In phosphorylation, there is an increase in mass of 80 Dalton, right? Similarly, methylation you can study, oxidation you can study. So in short, mass spectrometry definitely is used to identify the post-translational modification like acetylation. Why not other technique? So STS page cannot be used because it cannot differentiate between non-acetylation and acetylated protein. Light scattering, again, it is used to determine the molecular weight, not post-translation modification. And circular dichroism spectroscopy again cannot be the answer, because circular dichroism helps us study the secondary structure of the protein. So this question, for this question, answer is option 1. Then a transmembrane receptor protein X and transmembrane protein bound to actin cytoskeleton with fluorescent tags were expressed in a cell. So I hope you all have studied about membrane proteins, receptor proteins in unit 2, but it is related to this FRAP experiment, fluorescence recovery after photobleaching. So fluorescence recovery after photobleaching of these proteins were performed on these proteins. And the question is, which one of the following options represents, like it has been asked in the question, that which one of the following options represents most likely outcome of the experiment. So obviously we have to read the outcome, like we have to read the options for this. So first is, fluorescence of both protein will be recovered at the same rate. No, it cannot. It is not possible, because see, fluorescence recovery after photobleaching will be faster for those proteins which are mobile. But here in this case, we have a protein X, right? One is protein X, a receptor protein, transmembrane receptor protein, and another is Y, which is bound to actin cytoskeleton. So that means one is mobile, another is immobile. So definitely we will not see the recovery of fluorescence at same rate. This is not correct. Then fluorescence X will be recovered later. No, this is again incorrect, because X is mobile. It will be, you can easily, its fluorescence will be recovered faster than Y. Or you can say that fluorescence of Y will be recovered later than X. Right? I hope you all are following. So mobility of membrane protein varies, especially if one is cytoskeleton bound. So Y fluorescence will recover later than X, as it is bound to cytoskeleton, making it less mobile, leading to slower fluorescence recovery. And no recovery of fluorescence is expected. This is again incorrect, because one is free. X is free. So answer here is option 3. That represents the most likely outcome of the experiment. Then which of the following enzyme is used for conversion of blood glucose to gluconolactone in commercially available blood glucometer? So if you see the trend, now CSIR NET is framing questions on very basic instruments that we are using in our day-to-day lives, right? Like oximeter, glucometer, right? So if you remember the previous year, the question paper, the question was on oximeter. Now this time it is on glucometer. So these basic instruments working you need to be familiar with. So basically this is the reaction happens in glucometer, and it is catalyzed by glucose oxidase. So this gluconolactone, it is which has been produced through this reaction, is hydrolyzed to gluconic acid, and that generate hydrogen peroxide, and the generated hydrogen peroxide is detected electrochemically to measure glucose level. So here answer is option 1. Now, very interesting question. Every year there is a question on PCR, and the question based on the primers, forward and reverse primer binding to the PCR sequence, to the sequence, DNA sequence given. So question is, select the correct set of octamer primer pair to PCR amplify primer pair to PCR amplify DNA fragment containing the regions shown in uppercase letters below. Now, a very simple concept here you have to recall, and that is that in PCR, forward primer, okay, forward primer, it always binds to antisense strands 3' to 5'. I think, just a minute. So forward primer binds to 3' to 5'. I think just you all can note down, okay? My marker is little bit not working. Yeah. And reverse primer, it binds to your sense strand, that is 5' to 3'. So forward primer binds to antisense strand, and reverse primer binds to the sense strand 5' to 3' to initiate DNA amplification. So one thing is very clear. It is mentioned that this uppercase letter, okay, we have to choose the primer pair which can amplify this DNA fragment written in uppercase letters when you perform PCR. So one thing is very much clear, like only 5' to 3' sequence is given. So obviously we can write down the complementary strand sequence. So you have to write down the complementary strand sequence, and you don't have to write the entire sequence, okay? Because you know that primer will bind like only to the, because this sequence has to be amplified. So obviously primer will bind few nucleotides before this sequence, right? So obviously in 3' to 5' it will bind, and it will bind like over here. So point is that 3' to 5' sequence, whatever it is, the primer sequence will be exactly as given for like will be exactly having the sequence which is given over here, right? Because see, we are 3' to 5', there will be a complementary strand, and that forward primer binds to 3' to 5' sequence. So it will be exactly this one. Now this sequence A, G, G, A, C, T, T, A is given in option 1 also, option 3 also, and in option 4 also. So that means option 2 is not the answer for sure. Now the choice of option between 1, 3 and 4 will depend on reverse primer sequence. So reverse primer sequence will be complementary to this. Now don't forget, it will be like you have to read. This is 5' to 3'. So your primer will bind like this, 5' to 3', right? So while you are reading, it should have a like here you have to keep in mind the direction, right? So I have already written here. So you can see G pairs with C, G pairs with C, C pairs with G, G pairs with C, A pairs with T, A pairs with T, G pairs with C, T pairs with A. And this sequence, if you read it from 5' to 3', it is given in option 3, A, C, T, T, G, G, C, C, and therefore answer will be option 3, not 1 and not 4. So this is how you need to solve these type of questions. And then we have one more question, part B question. Sheep RBCs, SRBCs were added in wells 1 to 10 of a microtiter plate as shown in the figure below. Next, serum from a mouse that has been immunized with sheep RBCs was diluted 1/50 in a tube and then added in serial twofold dilutions from well 1 to 9. And you can see an image they have also given. And you have to find out which of the following statement is correct about titer of anti-serum RBCs antibody in the serum sample. Now, basically, remember, this question is based on hemoagglutination. So, hemoagglutination is a clumping or aggregation of RBCs, which are being caused by interaction of the antibodies with particular antigen. So it is often used in viral detection and diagnosis. And remember that this assay uses red blood cells as source of antigens. Now, to determine antibody titer of anti-sheep RBCs, anti-sheep RBCs antibody, we need to identify the highest dilution with hemagglutination, which is usually observed as a uniform diffuse red color in the well. So if you see the image given, we see a very uniform and red color over here. So up to 1 to 3 well it is seen, but not 4 to 10, right? And as it is mentioned in the question that the sheep RBCs were dilute 1/15 tube and added in twofold dilution. So that means in this well, it is added 1/100. Here it is added 1/200, and here it is added 1/400. And beyond this, we are not seeing the hemagglutination. So that means the titer is 1/400, and hence will be the answer. Again, very easy question. So these were the part B questions. Now moving over to the part C questions. So part C, I will be discussing only unit 9 and 13 questions of both morning shift and evening shift. So part B, we have already done. Now moving over to unit 9, part C questions first. So question is, table given below provides a comprehensive list of selected plant diseases and possible causal pathogens. Now remember, every year there is a question on either plant diseases and the causative pathogens, or animal or human diseases and pathogens, or even the vectors they sometimes mention. Now every time we always whenever we talk about the tips and tricks to solve the questions, we always say that you should go for match the following type questions first, because they're very scoring. And this time there were a lot of match the following type questions from unit 9, especially in part C. So even if you don't know about all four, at least two, if you are familiar, you can easily answer this question. And even in the regular classes, I always say that you should at least learn most common diseases. Like fire blight is one very common. Ergot of cereals is I'm sure you all have heard about it, right? So if not, then any two, whichever you are familiar with, you can you can use it to solve the question. So basically here, this is a very direct question, memory based question. You simply have to recall here what all plant disease is caused by which all pathogen. So if you see Venturia, it causes the apple scab disease, so A is 4. Ergot of cereal is caused by claviceps, so B is 1, right? And fire blight is caused by the Arvenia, right? So you have C2, and root knot is caused by the Miladogy. So it is D is 3, and A4 I have already told you. So this combination is given in option. So A4, B1, C2 and D3 is given in option 4, and hence it is the answer. Then match the animal in column X with its characteristic in column Y. So even if you have, like, this is a very general knowledge based question, right? I'm sure you all have heard of echolocation, right? Sonic wave, sonic, the signals that are being used, especially by dolphins. We even in some movies it has been shown, right? And even like maybe lateral lines, or any, again here, if any two you are familiar with, you would have answered it correctly. So again, it's a very easy question. So heat sensing pits are in snakes. So we have A is 2. So these are facial openings that contain membranes that detect infrared radiation from warm bodies. So these pits help snakes hunt in the dark by allowing them to see an infrared image of their prey. Then we have spinneret, which is a silk spinning organ of a spider or larva of insect. So that means B is 4 here. And lateral line is present in fishes and dolphins. They have echolocation. So lateral line is a sensory system that allow fishes to detect the weak wave, water motions and pressure gradients. So C is 1. And dolphins, they use sonar, like echolocation, also called as biosonar. They use it for navigation, hunt and detect dangers. So D is 3. So this combination, basically A2, B4. So see, A2 is given in option 1 and 4. So if you are familiar with this thorough with A and 2, then definitely you can omit 2 options out of 4, right? So this way also you can solve this question. So here definitely answer is A2, because B3 is given in option 4, but actually B4 is correct. And then we have C1 and D3. So answer is option 1 here. Then we have another question. The table below lists selected bird species and possible habitats. So as I told you either question earlier, that as the question on migratory birds or the natural habitats questions do come. So here is one such question. So again, if you are familiar with any two, you would have answered it correctly. So if you see this Bugun Lyocicla, it's a critically endangered songbird, and you find it in northeast India. To be more precise, it is found in Eagle Nest Wildlife Sanctuary within Arunachal Pradesh. So this is A is 2. Then grey-headed bulbul, you find it in Western Ghats. It's a member of bulbul family. You find it in, it is endemic to Western Ghats and southwestern India, and found from Goa south to Tamil Nadu. So B is 3. So A2 is given only in option 4. And further, B3 confirms our answer. And definitely the last one is Brooks leaf warbler. That means it is 1. That is, it is found in higher altitude of Western Himalayas. So it is the Western Himalayas are the endemic bird area. The answer here is option 4. Then table below lists food reserves in algal groups. So I'm sure if you have studied about algal groups, you would have answered it correctly. So there are many algal groups, only few are mentioned here. So I will be providing information about these 4 only. So basically paramylon, it's a carbohydrate similar to starch and it is a primary food reserve in Euglenopiceae. So that means A is 4, and A4 is given only in option 2. But we can further confirm our answer. So laminarin, or okay, let's talk about Caryophyceae. So that stores starch. So starch is stored by Caryophyceae, that is B is 3. So yes, this confirms our answer. Answer is option 2. And for your information, laminarin, it is reserve products in Phaeophyceae, that is C is 2. And then we have chrysolaminarin, which is in food reserve in Bacillariophyceae. The answer here is option 2. Then following table shows a list of migratory birds coming to India. Now see, this is a question on migratory birds. Black-tailed godwit, comb duck, ruff, and spotted redshank. Very direct, memory-based question. How many of you have done it correctly? Can I know it in chat? Please let me know in chat how many of you have done it correctly. It's a general knowledge-based question also, you can say. So black-tailed godwit, they spend their summers in Iceland or Russia, where they breed, eat, and raise their young. So definitely A is 2. Now A2 is given in option 2 and 4. Right? So that means answer cannot be option 1 because A3 is given, and obviously third option is not correct because A1 combination is given, which is incorrect. Then definitely like B3 is common in both and in option 2 and 4. So that means either you can answer this question either by looking for C or D. So comb duck, it is also called as natka, and it is called in India, and it is basically found in Madagascar and South Asia, and large freshwater wetlands in sub-Saharan Africa, Madagascar, and South Asia. But B3 is given in both, so we check for C, which is ruffs. So ruffs are birds of Arctic tundra region. So that means C is 1, which is given in option 2 and not in option 4. So that means 4 is also not correct. Answer is option 2. And last is spotted redshank. So it is in this Scandinavia. So answer is option 2 here. Then the table below lists structural modifications found in various plant genera. Phylloclade, cladode, phylloid, inflated petiole. So you should know what is phylloclade, what is cladode, what is phylloid, what is inflated petiole. Okay, so I have mentioned here, you can refer to the screen. Phylloclade is a modified stem, and these are mainly found in like Euphoria, then cladodes in Apantia, and phylloids in Acacia. So based on this, A2, B4, C1, so only D is left. And so A2, B4. So A2, on basis of A2, you can omit 2 options out of 4. Answer cannot be option 1 and 4 here. And definitely it will be 2 and between 2 and 3. So when I say B is 4, so that means answer is option 3. So C is 1 and D is 3. That is inflated petiole is in Pistia. Now coming to unit 13 part C questions. I'm sure you all must be waiting for unit 13. So as every year, we get direct questions from unit 13, combination based questions from unit 13, right? So this time the technique on which the question was based was EMSA, electrophoretic mobility shift assay. So this is one of the technique from where the experimental based question was asked. So definitely to answer this question, you should know what is this technique, electrophoretic mobility shift assay. So remember, EMSA, it is basically being used to study the protein and nucleic acid interaction. So I think there is, I'm not able to write it properly. You all can, there is some technical issue, I think. You can just note it down, okay? Protein and nucleic acid interaction. Yeah. So to study the protein nucleic acid interaction, EMSA technique is used. And always remember, when this complex is formed, it migrates slowly than unbound DNA. So to answer this question, it's an experimental based question where it is mentioned that two newly identified protein X and Y are tested for sequence specific DNA binding activity. The results of EMSA with a labeled DNA fragment and protein X and Y in various combinations are shown below. So here you can see the result is also given, the profile, the band pattern is given, and also the conditions like when these bands are obtained is also given, like when you add the X or Y protein like that. And so along with protein X and Y, poly, that is the DNA duplex of polyinosine and polycytosine is also added, okay, is also used here. So if we look at the table given and this the band pattern which is given, so you will see that free DNA, for free DNA, this is the bands. Excuse me. Now, so in first lane, I'll just label it as first lane. So just have a look at the table and this band pattern carefully. Accordingly, we have to solve this question. So if you see the lane 1, no X, no Y, and no this DNA duplex is added. Only DNA is added. So yes, we are getting one band. Now in second lane, if you see, in second lane, we have added DNA along with X. So can you see a shift? Basically, it is also called as EMS is also called as gel shift assay, because as I told you, the protein nucleic acid, if it is formed, it will migrate slowly. So we very well know that when you perform electrophoresis, the higher molecular weight molecules will move slowly and lower molecular weight move faster. So since complex is formed here, definitely it will be of high molecular weight than free DNA. So it will move slowly. So band we are getting over here. So this indicates that yes, X is binding to our DNA. Protein X is binding to our DNA. Then in third lane, we are adding DNA and we have not added X protein, but Y protein is added. So again, we are getting a band, and you can see it is at different position, right? You can see it is little bit down to a lower side than this band. Then in fourth lane, if you see, we are adding DNA also, X also, and Y also. So you can see we are getting a band corresponding to Y as we have got here, also X as we have got here, and we are getting one more band. So that means yes, both X and Y are they are individually interacting also, and they both are binding to DNA also. So both X and Y are also binding to the DNA. So that is why this band we are getting. Now, if you see the lane 5, in lane 5, we are adding there is a DNA, there is an X, and then we have added this another DNA duplex of polyinosine and polycytosine. So in that case, you can see that there is no band over here. No shift is seen. So that means X is not sequence specific. In presence of another sequence, it will bind to it. It is not binding to our DNA of interest. And if you see the last lane, that is sixth lane, you can see that we have adding Y protein, X is not added, and also this this DNA duplex is added of polyinosine and polycytosine. But we are getting a band here. So that means Y binds and it is sequence specific. So now we will look at the options and we have to find the correct interpretation. We have to find the option which represents correct interpretation of results obtained. So both X and Y are sequence specific. This is incorrect. I told you X is not sequence specific. In lane 5, we have seen that when this complex DNA duplex is added, X is not binding to this DNA, right? No band we are able to see here, right? Then X does not bind to DNA and Y bind to specific sequence. Now this is incorrect because X, yes, it binds. We have seen in lane 2, it do binds, right? So this is incorrect. Then both protein bind to DNA. Yes, X also binds, Y also binds. But Y binds in sequence specific manner. This is correct. We have seen in lane 6. So this could be the answer. And X compete with Y to bind the same sequence? No, this is not the case, right? Because when we have added both X and Y, we are getting individual bands also, and also we are getting one additional band which is indicating X and Y both binds to the DNA. So answer here is option 4. Answer here is option 3. Okay, 4 is incorrect. So both protein binds to DNA, but Y binds in a sequence specific manner. Now coming to another question, very interesting question. It is based on PCR and like how the amplification will happen. And this is a combination based question where you also need to have an idea about genetics, unitate, like what is heterozygous term means, what is homozygous term means. So the question is, a student used 4 20-mer oligos to amplify DNA using a regular tag DNA polymerase from wild type, homozygous mutant having a deletion of the gene. Okay. And so wild type was used. Okay, so you all can write down. Okay, I'll just speak because again. So wild type, let's say we represent with this allele. Okay. And heterozygous condition. Let's say represented with this allele. So here uppercase letter A allele represents normal allele and lowercase letter A represents the mutant allele. So mutant is which is with the deletion. Okay. And so last is mutant, which we represent with this condition. Right. Now, agrosgel electrophoresis profile using all 4 primers simultaneously on each template as shown below. Which one of the options given below represents the correct profile? Okay, so here basically if you see, these are the 4 options. So if you see, like first we analyze like what all primers are used for amplification. So if you see, this is the primer FW, right? It is binding over here. And then we have a primer D. So that means if these 2 primer combination is used, we will have the amplification of 500 and this 500, right? If we are talking about normal allele. So in that case, in case of normal allele, one band, we will get of 1000 base pair. Like we will get a band of 1000 base pair, right? And if we use primer FD, and primer D, we get only this portion amplified. So this is in case of wild type, when you don't have a deletion happening, we have a normal allele. So we will get 2 bands, one of 1000, another of 500, right? So based on the given options, if you see, we are getting, like you just have to check the lane for wild type. So if you see the options, then here you can see we are getting 1000 base pair here also and 500 here also. Then here also we are getting 1000 and 500. Here we are getting one more. So definitely this will not be the answer. And for option 4, we are getting one band over here and one band over here. Now for heterozygous, so for heterozygous, for wild type allele, we should get these 2 bands, 1000 also, and for 500 also. And one more thing is there. That if you are using primer FD and like primer R, right? So that also you have to see, like what is the combination, like what all bands you will get. So especially when you're talking about deletion region, right? So please correct, when there is a deletion region, first we'll talk about deletion region and then we'll talk about heterozygous. So if there would have been a deletion region, obviously this primer will not bind, this FD will not bind, and this D will also not bind. So you will have only primer R and you have primer FW. So that means you will get 800 band, because 500, this 500 and this 300. So approximately 800 band you will get, right? And for heterozygous, because it has both the alleles, so you will get a band of 500 also, 1000 also, and 800 also. So that means only single band you should get for deletion condition, right? Homozygous mutant. So if you see the option, in option 1, option 3, we have already omitted. Option 4, we are getting 2 bands, so this is incorrect. So we are left with only option 2. So this is correct. 1 band for which is around like if you see this is 750. So this is like around little bit beyond 750. So around approximately 800. And for heterozygous, we should get 3 bands. So this is the answer. Option 2 will be the answer. So same I have mentioned here. Okay, you can note it down, that what all combination of the primers we will be going to use, and you have to consider all the 3 conditions here. Okay, wild type, heterozygous, and homozygous mutant. So obviously, if it is a homozygous mutant, then definitely this deletion region will be there. So primer FD and primer D will not bind. Amplification will just be on basis of primer FW, which will amplify 500, and primer R, which is 300, because this entire portion is deleted. Then moving over to another question. Now this time there was a question on transcranial magnetic stimulation of the brain, and I know many of you might be studying it for the first time. So here I have added few information about transcranial magnetic stimulation. So the very first statement was that, so the question was that magnetic field generated from an electromagnet is used in transcranial magnetic stimulation of the brain. The following statement suggests some features of TMS. So features of TMS are given, and you have to find out that which one of the following options represents the combination of all correct statements. So basically, if you see here, the very first statement is magnetic field generated in TMS induces an electric field in underlying brain area. Now, definitely to answer this question, you should know what is this technique TMS. So for your information, you can note down, if you are studying it for the first time or hearing about, if you are if you have never heard it before, so you can note it down right now, that TMS basically it is a technique which is used to study the brain, and it works on principle of electromagnetic induction, where you use a magnetic field which in turn generates an electric field in the underlying brain area. So yes, A statement is correct. Then electric field in brain area alters membrane potential of neurons in the locality, causing them to depolarize synchronously, which in turn may change the probability of firing of neuron. So this statement is also correct. So here is the information about it, the detailed information about the principle of working principle of this technique. So yes, it passes a long-lasting current through the coil TMS and generates a time-varying electromagnetic field perpendicular to the transducing coil, which is placed tangentially on the head. So there are electrodes which are being placed, and the coils are being used in the instrument. So magnetic field is provided to generate an electric field in the underlying area, and the electric field which is being induced in the targeted tissue can depolarize excitable structures within the brain. So what are the excitable structures? Like neurons. And this electric field induced is sufficient to strongly depolarize the membrane potential of given neuron, and action potential will be triggered. And some more details are there, but just to make you familiar with this concept, I have added few details. Then in cognitive neuroscience research, TMS may be used as tool to induce a virtual lesion. This is also correct. In order to understand the function and temporary virtual lesion of a target cortical area is done to disrupt its function and associated behavior, to study it, to understand the function role of specific brain areas. And lastly, is TMS is safe and non-invasive, so it is correct. But another part of statement is that neuronal activity of stimulated area is disrupted for long period of time. This is incorrect, because if we are using a technique which can disrupt the, like you are gone for a treatment and or let's say for the diagnosis you are using this technique is used, and you are end up getting your brain activity for long period of time. So that is not good, right? It is not safe. So this statement is incorrect. Disruption is there, but it is short lived. Neural activity is returned to normal soon after the stimulation. So answer here is option, like combination of statements has been asked here, and correct statements, so it is A, B and C. D is incorrect. Now this time there were two questions, both in morning shift, and like one question in morning shift and evening shift. Like two questions were on SDS page, and I have added both the questions. And if you see here, the question was same. The only difference was in the this gel profile which is given, right? So if you see this questionnaire of morning shift, the question is 150 kilodalton protein species from gel filtration column was run on 2-dimensional SDS page. What is the likely form of 150 kilodalton protein species from this observation? Now, definitely to answer this question, you should know what is the role of SDS and what is the role of dithiothritol, that is DTT. SDS, so first of all, page stands for polyacrylamide gel electrophoresis. And SDS is an anionic detergent which is being used to linearize the protein to provide a uniform negative charge to the protein so that proteins are separated on basis of molecular weight. And also it provides, like it breaks the non-covalent interactions. Okay, so that the protein, it linearizes and it provides a uniform negative charge to the protein. So let's say we have added us protein and protein is separated. Now, if you carefully look this gel profile, you will find that the bands we are seeing are in the same lane. They are not in the different lane. Okay. So first the electrophoresis was done in this direction. And then further SDS page was being done by adding dithiothritol, which is a reducing agent. It breaks the disulfide bond. So if we look at the options are that there are at least two proteins that are linked through non-covalent interactions or covalent interactions. So these two are contradictory. So obviously, what do you think? When you are adding SDS page, right? Now just think about it. When you are adding, when the sample is run as a, when it is run simply performing SDS page, it is running as a single 150 kilodalton protein. That means it is acting as a, behaving as a single species. It's only when you add a reducing agent, you are getting two bands. So if there would have been a non-covalent interaction, SDS alone would have dissociated it, and we would have seen bands, two bands, but in different lanes, right? But since here we are getting the bands in same lane, so let's say one must be of 100 kilodalton, another of 50, or let's say 2 of 50 or 2 of 25. This way also you can write it as, okay, like 2 of 50 it can be, or it can be like 2 of 25, right? So obviously the answer here will be, what do you think? Covalent bonds, because non-covalent bond, if they would have been, then they would have dissociated and we would have seen bands in two different lanes. But since the band is in same lane, so definitely there are like two protein, like the protein which is connected through a disulfide bond. And this disulfide bond, it breaks on adding the dithiothritol, and therefore you are getting the same lane, you are getting the two bands. So answer here is option 2. Okay. And it cannot be correct, like two proteins in mixture without forming a complex, or one protein that has disulfide bond. No. The similar question was asked in evening shift, but here you can see that bands are in different lane, right? So just now I have already explained you two bands, like for example, first time when the electrophoresis SDS page is done, so your protein is separated. Okay, so it is connected through non-covalent interaction. So let's say they have migrated this much. Now further, when you perform the SDS page with dithiothritol, so this protein migrated up to here and this protein migrated up to here. So you can see one is of higher molecular weight and other is of low molecular weight. So either this could be of 100 and this could be of 50, right? So here, because we have got, let's say, two bands in different lanes, it means they are linked through non-covalent interaction. So therefore, answer is option 1. Now you may say that, ma'am, there is a possibility that even instead of 50, there must be the protein which is connected to disulfide bond also, right? One of 25, another of 25, which further breaks on using dithiotretol, right? So yes, there is a possibility. You are absolutely right. It is possible. But if you look at the option 2, it is given at least 2 proteins in the complex. So no, 2 proteins up to this is fine, but at least 2 proteins, that means um, this particular term doesn't go well here. Okay, maybe 2 proteins is fine, but at least 2 protein, this is not correct. So that is why answer here is option 1 and not 2. So everybody, I will just take 2 more questions, okay, because it's almost 8 o'clock. We have already exceeded the time. So quickly moving over to this question, it is direct based on ELISA technique. We all know that ELISA technique is used to detect a specific antigen or antibody in the sample. So here this is an experimental based question where a student performed an ELISA to detect anti-oval albumin IgG in serum sample. The experiment involved in following sequential steps: coating plate with ovalbumin, blocking with BSA, adding serum sample, adding anti-mouse IgG HRP, adding H2O2 plus OPD, and adding H2SO4. Now you should know how this technique is done. Like to perform ELISA, like you should be familiar with ELISA technique and you should know what is the role of OPD, what is the role of H2SO4, then only you can answer this question correctly. And here in this question, we have to find the option which represents a combination of all correct statements. So if you see option A, if the plates are not blocked with BSA, the specificity of assay decreases. So we have to choose the correct statement. Okay. Now, if the plates are not blocked with BSA, bovine serum albumin, and other reagents we usually use, BSA is often used to been used to block the plate. So the question is that specificity of assay decreases if the plates are not blocked. So yes, it is right. This is correct. That is why BSA is added, so that non-specific binding of antibody doesn't happen or antigen doesn't happen onto the plate. Specificity, sensitivity is very important when you are performing an experiment. Then statement B is if plates are not washed between addition of serum sample and addition of anti-mouse IgG HRP, the sensitivity of assay decreases. Correct. Because if the washing is not done, then maybe unbound reagents still be there and it will reduce the specificity, sensitivity of the assay. So this is correct. Then if the plates are not washed between addition of anti-mouse IgG HRP and H2O2 plus OPD, the specificity of assay decreases. This is also correct. See, washing is required after each step that we performed in ELISA to remove unbound reagents, be it antigen or antibody from the plate. So C is also correct. Then OPD is the substrate for enzyme. Now this is incorrect. Kindly note OPD. Yes, there are, like, if you read about the substrates of ELISA, in some places OPD, yes, it is a substrate which is being used. But here you have to remember that, so OPD is a substrate for HRP, but horseradish peroxidase, but you have to remember that here we are adding H2O2 also. So remember, when H2O2 is also being added, okay, so H2O2 substrate act as a substrate, because HRP uses it to oxidize molecules like OPD, which generates colored product, which can be detected. So this is incorrect. And E is without H2SO4, no color is developed. This is incorrect because H2SO4 is just used as a terminator of the reaction. So the correct statement is A, B, and C, which is given in option 3. So answer here is option 3. Then this is a very interesting question. You have to just calculate mean median mode here. So question is given below are leaf lengths. Leaf lengths measured from a sample of 15 dipterocarp trees. What are the mean, median, and mode of leaf lengths? So what is median? What is mean? Mean is the average. So what you need to do is, first you have to check whether all the the values in the data set are in ascending order. So it is already there. How many total values are there? 15. Now what is mean? Mean is the average, right? So how will you calculate the average? Sum of all the values in the data set divided by total number of, like, total, you will add all the numbers of data and divided by the values in the data set, right? So there are total 15 values. And when you add them all, you get 134. So 134 by 15 is 8.93. Median is the middle value. Now, if you see 15, so 15 is an odd set, odd data set it is. So middle value is what? The eighth number. If you take it from here or if you take it from here, the eighth one. Okay, so eighth one it will be. That is 9. And mode is the one value which is frequently occurring. So it is eighth, which is occurring 3 times. And therefore it is the answer. So where is this combination given? In option 2. Mean is 8.93, median is 9, and mode is 8. Now, this is question on flow cytometry and unit 4, combination based question. So question is, breast cancer stem cells may be identified by analyzing CD24 and CD44 phenotype of breast cancer. So CD24, CD44 stain cells were analyzed by flow cytometry, and further analysis of sorted cell from different quadrants Q1 to Q4 by immunoblotting revealed the following result. The dotted line represents lesions with negligible signal. So based on the results, which phenotype is associated with maximum number of breast cancer stem cells? Now kindly note, here you just have to recall the dot plot of the flow cytometer. We all very well know that Q3 is double negative always. That means this quadrant indicate absence of both CD24 and CD44, right? So absence of both, this indicates. Then quadrant 2, that is the upper right quadrant always is the double positive. So this will always indicate the presence of CD24 also and CD44 also. Okay. And if you see, so if you see quadrant 1, it indicates presence of CD44 but absence of CD24. And if you see quadrant 4, it indicates presence of CD24 but absence of the it indicates the presence of CD24 but absence of the CD44, right? So if you see this data which is given, so it is given like the dotted line here represents negligible signals. So basically whatever cells we are getting, on further analysis of them, following result was obtained. So you can see for HTERT and beta tubulin, we are more or less getting the same result. But if you see these for SOX2, OCT4, this OCT4 and NANOG, you will see that in quadrant 3 and 4, like negligible signal we are getting. And for quadrant 1 and 2, in these two, you can see that for more prominent bands we are getting for quadrant 1, right? And we know that this SOX2 and OCT4 or NANOG, OCT4 and NANOG, they are what? They indicate what? They all indicate the cancer cells, right? Like they are associated with the cancer cells, cancer tumor cells, and they are indicating the progression and the resistance, chemo resistance, right? So definitely if it number increases here, so that means which is associated with most number of breast cancer stem cells? It is quadrant 1. And quadrant 1, I told you, it is indicated with CD44 plus and CD24 minus, which is given in quadrant 3. So answer here is 3. Now this is a very easy question. It's just that instead of giving directly the values, they have given you a graph, right? A spectrum. And it's a question based on Beer-Lambert's law. So the question is, absorbance values of dyes measured at 600 nm was plotted. So as we all know, according to Beer-Lambert's law, absorbance is directly proportional to concentration. So different concentration of the dye is given, 3, 6, 9, 12, and so on. And absorbance is recorded. Like absorbance at 0.2, absorbance at 600 nm was recorded. So 0.2 corresponds to like 3 concentration and so on. Like we have 4 corresponds to 12, 0.6 corresponds to 18, right? Like that. So path length is given 1 centimeter, and you have to determine molar extension coefficient. So from Beer-Lambert's law, we know that absorbance is equals to mole extension coefficient, concentration, and path length, right? So this is the formula. And to determine mole extension coefficient, you all need to just do is that you have to write the value of absorbance, concentration, and path length. Now, concentration and absorbance you can get from the spectrum. So you can take any absorbance. Let's say I take 0.6. So when I take 0.6, concentration corresponding to 0.6 is 18. Now, kindly note it is in millimolar. And we have to determine the value in molar per centimeter, right? So, so 0.6. So 18 millimolar, if you convert into molar, it will be 0.018, right? And path length is 1. So when you solve it, you will get around 33.3. And therefore, the answer here is option 3. Right? So answer here is option. Very easy question. Another question, similar question was there, that molecular absorb light at X nm wavelength and emit lightest fluorescence at Y nm wavelength. Typically, there is a shift in the wavelength. So it's a red shift, you can see, because light emitted is of longer wavelength, right? So Y is more than X. So E is the energy transferred to solvent during recognition of excited state. H is Planck's constant and C is speed of light. Kindly note, always energy is equals to H. How will you determine the energy relation? So in physics also, I'm sure you all have studied that energy is equals to h nu, or you can write hc by lambda. Okay, so nu is the frequency, which is equals to c by lambda, velocity of light, speed of light by wavelength of light. So as here, energy of photons, so hc by lambda is determined. So if for two wavelength x and y, we can, so x refers to absorbance and y refers to fluorescence, so it is an emission one. So we can write the energy, which is hc by x. So because in the question it is mentioned, right, that we have the x nm wavelength which is absorbed and emitted is y, fluorescence at y. So one is absorbance, another is energy of emission. So energy difference we can calculate by energy absorbance minus energy emission. So if I put the values hc by x minus hc by y, it will correspond to this equation, and this is mentioned in equation 2, and hence it is the answer. And this is the last question for today. And again, every year such type of questions are asked in CSIR NET exam, and this year also the question was asked. And it was on the restriction enzymes, the combination of enzymes that you can use to generate the compatible ends for ligation. Now, one rule that you always have to remember is that blunt end, the enzyme which generate blunt end. So any blunt end you can join with any other blunt end. So if you look at the given restriction enzyme, so you have to just go through the restriction enzyme, what kind of ends they generate. So EcoR5 generate blunt end. And if you see the AVA1, it generates the sticky end. HIND3 also generates sticky end. SAL1 also generates sticky end. SMA1 generate blunt end. And XPA generate sticky end. So that means anywhere where this combination EcoR5 and SMA1 is given, that can be used. Now, if we look at the option 1, so SMA1 and EcoR5 is given here. Okay, fine. In option 2, SMA1 is given with XBA1. Now, XBA1 is generating sticky end. How can you join let's say here it's a vector which is cut with the SMA1, so blunt end is there, and XBA with a sticky end, insert with a sticky end. Not possible. So this cannot be the answer, right? Now, in option 2, there is different combination of the enzyme which generates sticky end is given. And in option 4 also, EcoR5 and SMA1 is given. Now, definitely this EcoR5 and SMA1 can be used. But now you can check for compatibility HIND3 SAL1, or you can check for AVA1, like even HIND3 and SAL1 is given here also. So you can check the complementarity. Compatibility means here you have to check that whether the ends generated by HIND3 are complementary to the end generated by SAL1. And same applies here. Okay. So I have compared the AVA1 and I have compared this SAL1. So remember, the sequences by default which are given over here are always 5' to 3'. And this mark, the symbol, it indicates where the enzyme will make the site where the restriction enzyme will cut the sequence, right? So if you are considering the AVA1 5 prime to 3 prime, for SAL1 you have to compare 3 prime to 5 prime sequence or vice versa. Okay. So here I have written 5 prime to 3 prime, C, Y, C, G, R, G, exact same sequence. SAL1 given is again 5 prime to 3 prime. So I will write its 3 prime to 5 prime sequence, which I have written here. Okay. Y corresponds to any pyrimidine. Okay. And G corresponds to any A or G, purine. So obviously, if so, this is SAL1 this 3 prime to 5 prime sequence, and if you just need the 4 base pair complementarity, that is after where the cut is being made. Okay. So if you see for AVA1, the enzyme digest here, and over here it digests. So you can see A pairs with Y, so Y can be any C or T. So if T, it binds. G pairs with C, G pairs with C, right? Or C pairs with G, you can say, and T pairs with R. R can be if it is adenine, then it's fine. So yes, this means answer is option 4. And even I tried checking Hind3 and Sal1 and they don't show complementarity. So on that basis, we can say answer here is option 4. So this is all for now. I hope you have enjoyed the session and you have identified where you have committed any mistake, if any, right? So I hope you now have learned what all strategies were being used to solve a particular question, what all concepts were used, and I hope now you have checked like what all answers, like whether the answer you have marked was correct or not. Okay. So if please let us know in the comments, and even you can mail us. You can let us know. Okay, you can call us on a toll free number and let us know how much marks you are getting, what are the chances of your what are the chances of qualifying CSIR net, and if any way, and like after discussing the question, if you feel like that you are not getting you're not scoring well, then don't get disheartened. Okay, remember CSIR net is a competitive, national level competitive examination, and it happens sometimes we do well, sometimes we don't, but instead of simply leaving the things, okay, simply quitting is not helpful, right? So you can start from scratch again, and in this, if you need any kind of help, remember Biotechnica is always there for you. Okay, so don't forget to let us know whatever, don't just let us know your you can share your feelings, you can share your scores. Okay, and if you need any kind of guidance or assistance, also for that you can always contact us. So with this, I end this session. Thank you, everybody, and take care.